All right. So can all of you see the PowerPoint now?
Yeah.
Yes, sir.
Cell signaling part 1?
Yes.
Okay. All right. So let's take a look at that. Here we are looking at the cell signaling. So remember that the cell surface receptor, the intracellular receptor, what are the basic differences in between that? Hydrophilic versus hydrophobic. These are some of the important considerations. Remember those things. Then let's write here. Different response in different target cell. So remember that what are the different type of response that can happen. That's important. And then this is a very important thing that you can absolutely expect questions that know what is a paracrine, short range, synaptic, long range, and endocrine long range signaling. And why we call this synaptic long range, because remember that not all synapses should be long, but you have some long neurons just going from your brain to a particular level of your spinal cord, which could be 3 feet long. So it can be a long range signaling as well. Autocrine signaling, remember that. So these are some of the hydrophobic signaling molecule, and remember that these are called the steroids because they all originate from the cholesterol. That's an important consideration, that these planar molecules are all from cholesterol, and that is important. All right, hydrophobic synthetic molecules. These are some of the just other names. So know the names, that what are the different ones in there. So here, just know that how a hydrophobic signaling molecules and intracellular receptor does work. So here, always remember when you are studying these questions, some of the questions can be asked that suppose this is a DNA binding domain, and if we just mutate this domain that DNA cannot bind, what will actually happen? Very simple, that if it cannot bind the DNA, then there will be no signaling. So because this is a transcription factor. On the other hand, think about that if there is a chaperone protein right here, HSP90, so suppose you think about that this is a ligand binding domain, and the ligand binding domain is mutated in such a way that it cannot bind to this HSP90 or the chaperone. Again, I'm repeating, suppose this ligand binding domain is mutated in such a way that it cannot bind, and the patient suffers from something intense. I'm just saying, just hypothetical disease, which is very, very detrimental to the patient is alive, but it suffers from something very, very detrimental. Say, for example, very high blood pressure, very very fast heart beating or something like that. So what is happening to that particular patient as well is that as it cannot block this particular association, this particular flexible region is on, and a small amount of these steroid hormones, which doesn't need to replace these HSP90, can readily go and bind in there. So you have to understand that the only way that it can change these DNA binding domain is that this is bound right here. That will change the DNA binding domain, and this particular ligand binding domain is interacting with this protein to keep this particular DNA binding domain separate. So there could be a condition that this is completely mutated in such a way that this DNA binding domain is completely exposed and ready to bind the DNA. So this gene is constantly on. And I will just tell that experiment that is happening, and you just have to just understand that the mechanism of that once this DNA binding domain is exposed, and that will lead to the transcription activation. So I will just give you a condition somehow that this is completely on because it is completely exposed by telling some kind of changes that is happening due to some kind of mutation. And again, just a basic concept of that. So this is the overview of the concept, that the primary response. So just remember that this is an important concept, that the primary and the secondary response. So remember that your primary response or the early genes are transcription factor that can do two things. It can have a feedback to minimize the initial response, And also it can turn on the secondary genes, means these are working as a transcription factor for the secondary genes. So these are some important concepts. So these are all good. So relay system, definitely know that how this signaling by phosphorylation happens or by the GTP binding happens. Basically the same, but the involvement of GTP exchange, that is important. Here you have an ATP phosphorylating the particular output which is active. So that's good. And again, the principle of the protein, that's good. So here, again, remember that the whole, you need to remember the G protein in a quite a bit of detail. So remember that this is all good. So here, this stimulatory G protein linked receptor and very clear understanding that how the hormone binds, and at that time your alpha beta subunit is associated with the gamma subunit, and how the GDP is bound to that in an inactive state, and upon ligand binding how it dissociates and the GDP is replaced by the GTP. So know the whole thing. This is very, very important as well. And know some of these FRET experiment, that how it will work. Now, when you are doing the FRET experiment, just remember that one of the thing that if you are looking at the fluorescence level as well, just follow the curve that sometimes the fluorescence level may not come down all the way down. So just make sure that there will be no trick. It will be a straightforward question. So do not overthink about some of these questions as such. If you see that the two molecules are interacting, you see a spike in the emission wavelength of the second one, or you do not see that. That's basically what it is. There will be no trick if we ask those type of questions. So definitely remove that. So here we are looking at, say, for example, fluorescence of the yellow light, and you can see that how fast the alpha subunit dissociate. And here you have the CAMP added and you have a decreasing fluorescence. Now, the question is that if you just do not get a like, you can see that it is going down, but it's not really zero. There is some signal out there. And these are the experiments, and you really will not see that in reality. That doesn't happen that when you do an experiment. So if it doesn't reach all the way down to the zero in there, that does not mean that the thing that what you are trying to see, that is not happening. It is dissociating, but some will not dissociate. That is absolutely fine. As long as you see a decrease or a trend, that experiment is good enough. That this is happening. When you add the signal, it will be a dissociation. That's good. So these type of concepts, remember that. So definitely remember that how the stimulatory and the inhibitory G protein works. And again, the important part is that when we went to the cholera toxin, we will come to that you will see that the effect of the cholera toxin and the pertussis toxin are the same. Because to work in entirely two different types of cell type that I just mentioned in the class. So the outcome is the same. And we'll just go into that in a second. Let's take a look. Let's open up the next one. All right, so right here, we are looking at the next portion, that how the signaling of the protein kinase A happens, binding to the regulatory subunit and separation. So know these, what are the outcomes? That's good. So this is very important. So here you have to do a little bit of memorization for sure, that what are the different kinases, phosphorylase kinase, then you have glycogen phosphorylase. So definitely remember the name of these enzymes. You have just 3 sets of enzyme and what is happening in each step. That is important. So here you are looking at that overall, the transcription by the G protein coupled receptor, and this is basically the overview of that. And here you're looking at the CREB-CBP complex, and the outcome is the transcription of that. If I do not think that I will ask CBP-P300 or CREB the detail of the names and individual what they're trying to do, as long as you know that this complex is basically the transcription factor, that's good enough. So this is very, very important. So definitely remember that what is happening in this process. So this is your cystic fibrosis structure, know that, and how it is activated and the role of this regulatory subunit. This is very, very important. That is different from what we learned beforehand. We did not go into the phosphorylation of this one, because remember that if this is not phosphorylated, the activation, deactivation doesn't happen. So that is very, very important as such, that how this happens. And then definitely know the overview of the process. This is the overview of the genetics of that, but the most important is that compared to that, we are not going to ask these questions, but these are also some of the overview that why this is so prevalent. But the main thing is that the mechanism, that how this is happening, and this is the main thing that you should focus on to. That in one case, the cholera toxin keep that G protein in constantly GTP bound state on. So that causes cyclic AMP to rise, protein kinase to rise, and CFTR activity to rise at that time. Large volume of water, extreme diarrhea. So this is what is happening with the cholera toxin. Because as you will secrete a huge amount of your chlorides, at that time, what will happen, it will pull a sodium chloride and it will pull the water due to the salt that will cause the diarrhea. So here you can see that the first thing is this high amount of cyclic AMP increase because it's constantly on. Now, important thing is that this is epithelial layer. When we are going into the next one, which is the pertussis, remember that here it is the same thing, but instead of your intestine, we are going into the lumen of the respiratory tract. So here what is happening, your pertussis toxin is GTP bound constantly off, but it is still making a high amount of CAMP. And this is one of the critical thinking questions. Remember that the outcome in the cholera, increased amount of CAMP and the consequences, and in pertussis, high amount of CAMP and the downstream is exactly the same. But what is the difference, that why it is off and CAMP is high? Remember that I just wrote that in the board. Think about that the particular adenylate cyclase in the respiratory tract works at a different pace than in the cholera. So than in the intestine. So here what is happening, that the adenylate cyclase, which makes the CAMP, suppose that works at 20% level. 20% is its basal level when it is not stimulated. When it's constantly stimulated, it is working at its 100% level. So think about that. Basal level 20% versus when it's constantly on, it is 100% at that time. So on the other hand, if you just go to your lung cell, go to your lung cell or the respiratory tract cell, here the adenylate cyclase, which makes the CAMP, normally working at its 100% level. So when there is no interaction of anything, no signal, it is working 100%. So when it is off at the GDP bound state at that time, it will basically will not be able to. So when the GI protein comes in at that time, due to the inhibitory signal, that 100% working will go down to say 20%. But as the GI protein or inhibitory protein is off, that 100% working is not coming down and it is working at the same level. That's the reason that CAMP is up. So one is doing from 20% and going up to 100%. On the other hand, in a normal condition, which is works at 100%, that needed to be down by these activated state to a particular state like a 20%, and that is not happening because this off GDP bound state is not allowing to do that. So the outcome is the same. So very important concept, and take a moment to understand that. All right, any other thing? Let's see, what is, sorry about that. Yeah, I mean, like if there's something going on, immediately tell me. My speakers and everything is on. All right, so here, what we are trying to see that again, know that the phospholipase C and how it does that IP3 signaling and the calcium signaling, that is very important. And here, what I said is that know that what is your PI, PIP, and PIP2, and how PIP2 is converted into DAG. which is also the diacylglycerol is also a signaling molecule, and IP3 is also a signaling molecule. And just remember that function of PI4 kinase and PIP5 kinase, and this is just kind of a few of your memorization type of questions. So just spend some time to understand that no structure is needed, just the name that what converts to what and which are the signaling molecule. Remember that DAG and IP3 in the whole process are the only two signaling molecules. And these are membrane-bound lipids. Remember that. These are in your phospholipid bilayer and signaling molecule, not your IP, but the DAG is associated still with the membrane even after it is formed. So remember that. So here, one of the important concepts is remember that this is very, very important, that outside the cell, if you look at the calcium concentration 10 to the power minus 3 molar, whereas inside the cell, the calcium concentration is 10 to the power minus 7 molar. So it is extremely low, and there is a reason for that. Remember that if you recap from the lecture, it is there, and you can just read the transcript as well. The calcium is an extremely powerful signaling molecule, and you need to keep the calcium level low inside the cytosol. Otherwise, it will randomly signal things. So to do that, you have two things that you can do. So you already know one thing, that when sodium comes into the cell, it's anti-pour the calcium, so it pumps out calcium outside the cell. So this is one of the way to maintain the low amount of calcium. Another thing is that calcium ATPase, that also help to keep that calcium amount low in the cell by pumping them actively. There are other two mechanisms. And so there you can easily see there's several mechanisms and know these things, that in the cell, on the cytosol, you have these calcium binding molecules, say for example, calmodulin, you have already heard about those. Once these are bound, these proteins are bound to the calcium, they will not be able to signal. So that's one of the way that the calcium, even if those few calcium molecules which are in the cytosol, they will be prevented from signaling once they are sequestered or bound by these calcium binding molecules. On the other hand, not only your endoplasmic reticulum, even some of the organelles will have these calcium ATPase, similar to that they have in the plasma membrane. So all the free calcium in a normal condition will go into these organelles, either in the endoplasmic reticulum or into the other. And these are all the processes. So you have 1, 2, 3, 4, 5 processes that are taking place to keep that calcium level low. So definitely remember that, so that you do not randomly signal. So here is an interesting thing, that how it is happening. GQ linked receptor. So we talked about the GS, which is the G stimulatory in the previous one, GI, G inhibitory, GQ produce IP3, and this Q is just random. But the principle is basically the same, that a GDP is replaced by the GTP, and then this will dissociate and activate the phospholipase C bound to the membrane, which will convert that into the IP3. This is one of the signaling molecule that will release calcium from the ER, modulate activity. Also, it will form DAG, which will activate protein kinase C, phosphorylation of cellular protein. So you can see that one signaling by the phospholipase C can signal in 2 different ways. So definitely remember that what are these 2 different ways. That's important, that it can do both, just not one. And here is the overview of that in a very specific way, that what downstream it does, that it releases calcium from the endoplasmic reticulum, as well as you can see that it also does activity. Once this calcium comes out, these intracellular calcium in the cytoplasm will also activate the protein kinase C, which is also activated with the help of these DAG molecules that will phosphorylate the substrate. This is one route. The next one, although we went into the detail of the structure, I'm not going to ask any of these names, STEAM, CAD, ORI. I'm not going to ask what is happening. Just remember that this is a store calcium operated channel. So again, I'm repeating, this is a store calcium operated channel. And what we mean by that, the only function that is the only part required. So here you are seeing that you have this IP3 gated calcium channel. So how does it work? You have this endoplasmic reticulum with all the calcium inside the endoplasmic reticulum. When this signaling cascade happens, So when this signaling cascade happens, the IP3 binds to a particular channel on the ER membrane that will release all this calcium. Now, suppose there is a constant signaling going on and there is not enough calcium in the endoplasmic reticulum. All of these cellular calcium are bind to the protein kinases and the cellular calcium is running deficient and still the cell need more. At that time, what happened is the activation of this one, which is called the STORE, which is the ER calcium operated calcium channel. Again, as I said, just to repeat the small thing right here, is that once this IP3 is bound to this ER membrane, it will release the calcium, and at one point of time, there may not be enough calcium in the cytosol and may not be enough calcium in the ER as well. The reserve is depleted. At that time, this will start. So this one is a STIM1 protein. As I said, the STIM1, CAD1 detail of the names are not required. But what is happening to this protein, that when you have high calcium in the ER lumen, this is bound to this, and you have a configuration like this, which does not interact with anything. This ORI1 is a calcium channel in the plasma membrane, not on the ER membrane. This is on the ER membrane, this is on the plasma membrane, different things. But when the calcium runs low, these bound calcium will get released, and at that time, there will be an oligomerization, means multiple of these subunits right here, multiple STIM1s, so you have 1, 2, 3 will come together, and they will insert themselves onto this ORI1. And the process of this activation is still not known. But what happens that once you have a low calcium, they just try to find areas where you can go into an area close to the plasma membrane, and this portion is inserted into the plasma membrane, where this will open up the calcium channel and the extracellular calcium will replenish them. So definitely the overview of the process. All right, so here there are some of these which are a little bit needed, how this signaling is happening right here. So one of those is the C kinase, a path with a MAP kinase I-kappa-B pathway. So once this DAG activates the protein kinase C, know that the MAP kinase will phosphorylate some of the transcription factor. On the other hand, the protein kinase C will also phosphorylate the I-kappa-B, which forms a dimerized heterodimer with the NF-kappa-B. Remember that NF-kappa-B is the transcription factor that needed to be released from the I-kappa-B, and it can only happen due to the phosphorylation. And that can just go down and bind to the transcription factor, lead to the mRNAs. So that's basically what it is. This is straightforward. Again, individual names are not required. Just know that what the main thing is, the receptor tyrosine kinase, that what we are going to talk about in general. So this is an important thing, this is in your book. So know the activation of that. So the important concept here, which is important, that upon ligand binding, there is an activation loop, and this is the one which, the activation loop is the one which first get phosphorylated. Once this get phosphorylated, the other side is autophosphorylated. Remember that the phosphorylation is not done by another enzyme, but the receptor tyrosine kinase phosphorylate all these tyrosine residues by itself. But it starts with the activation, this first loop, which is called the activation loop. Conformational change of this one is needed to activate its phosphorylation activity so that it can phosphorylate other, so it's in a very active state. All right, so we already know that how this RAS work, which is the GTP-GDP exchange activating protein. So definitely remember that. And that's a process a little bit of in detail. So remember that the function of what it is, that SH2, GRB1, SH3. So I highly suggest you to at least listen to the video, particularly for that. that how a sauce comes in and how this is the the interaction of the GRB2 and the SOS and how this actually happens and how the active RAS is separated out from there and the signaling happens. So pretty much the whole process. This is an important area. Some of the things that remember that we can ask sometimes that suppose you have a mutation of this sauce, and if you have a mutation of the sauce that I tell you, I supply the information that right here, the RAS cannot separate from the SOS. It cannot separate from the SOS because you have a mutation which cannot exchange the GDP to GTP. At that time, what will happen in a hypothetical state, which is basically there will be no signaling. If it is not replaced by the GTP and it cannot have the ability to separate, there will be no underground signaling as well. On the other hand, say for example, you have this SH2 or SH3 domain of the GRB2, and somehow I'm just telling you that the SH2, I will give you the information that this GRB2 do have this SH2 domain, which is bound to your receptor tyrosine kinase, and it has the SH3 domain, which binds to the SOS, and in a particular mutant form, this SH3 is mutated so that it cannot form a bond with the SOS. It doesn't bind. At that time, what will happen? So at that time, also the same way that if the sauce cannot bind, which is the nucleotide exchange factor, if it cannot bind to this SH3, then it will not be able to exchange the nucleotide. So this step will not happen, no signaling. So these are some of the things that this is known that the SOS is the RAS nucleotide exchange factor, or GEF, or the guanine nucleotide exchange factor. So again, we talked about that interaction of these proteins will happen. So when these proteins interact, you can track them with the fluorescence energy transfer. Interaction of these protein can be transferred. And just understand this experiment, that how it is being done. You have a complex molecule where you have the YFP on one end, CFP on one end, and then you have RAS and RAF together. So when there is a condition when they are separate, you can see that if you activate the CFP with a 433 nanometer, it will emit 475. But when in an activated form, when the RAS and RAF will bind together, where the GTP, the exchange of the GEF will happen at that time. When it is bound form, which is a RAS is a GTP bound form, this is the active form. So in the active form, it will bind to RAF. That's the important part. So once it's bound to RAF in the GTP bound form, YFP, CFP will show FRET. So you excited with 433 and it will emit at 527. Remember that. So if you're just interested, I'm not going to ask that farnesyl moiety is nothing but a portion which helps it to insert into the membrane. That's it. So that's the basic idea. So, but that is not needed. But the important part is that see that if the gap hydrolyzes the GTP to GDP, at that time, they will fall apart and there will be no FRET. Whereas if it is exchanged, the GTP exchanged to the GDP with a guanine nucleotide exchange factor, they will show FRET, otherwise it will not. So just know the experiment. Seen that experiment, basically. So this is good. And here also a good number of a little bit, and these are some of the things that you can just write down and that go step by step, that how this 1433, that is activating the RAF and how the RAF activates the MEC. And these are some of the very basic, very important protein signaling, like a signaling pathway. That's the reason you need a little bit of memorization for these. And that's what it is. But there, if you just compare to the book, that how much it is substantial, but again, it is not too much. So just know that how the dimeric MAP kinase activate this particular transcription factor in 2 steps. So see that right here, what is happening, that TCF is being phosphorylated by this MAP kinase right here, and that is not sufficient. you need what is called a tripartite. You have three factors where you need this phosphorylated SRF into which is phosphorylated by another protein, which is active, this P90 RSK. So MAP kinase activates the P90 RSK by phosphorylation. It also activates this TCF in the nucleus. Now, your active P90 RSK activates this SRF, and two SRF and one TCF binds together to this serum response element, which will essentially turn on the CFOS expression and cell change. So what I will suggest that you can just use just a blank paper to write these steps quickly, and that's probably the best way to study these steps. You need a little bit of memorization here for sure. And again, we are not going to ask that. This is a multiple different MAP kinase pathway just to show you, not needed. I'm not going to ask these details. All right, just a little bit of adaptation. Know the two process that what happens. One is desensitization, where there is a lysosomal degradation that can happen. And another is that very basic way, in your book, it is in much more detail that you can easily see that receptor phosphorylation by a particular kinase can lead to a condition where it can be bound by a protein right here, say for example, beta-arrestin, that will desensitize the protein. So just the two methods, one is a phosphorylation method, one is a degradation method for desensitization. So that's your 3.1 pretty much. Let's take a look at what we did here. All right, cell cycle part 1. These are fresh, so I think that you guys are good with this. Know the different phases. Of course, this is your mitosis. Just brush those ideas that what is happening. Very important. So just make sure that you know the function of the kinetochore, the astral microtubule, kinetochore microtubule, and the polar microtubule, the concept of that. And remember that all the minus ends are towards the mTOC or the mitotic right here to the spindle poles, and it's radiating out the pluses. And know that how this binds and the bioorientation is possible. That overview of that, right? Of course, not needed. And know the two method of search and capture, that it can just bind with the end or from the side. And a little bit of right here, just one take-home message from here, that remember that when these two sister chromatids are pretty much trying to get into the middle portion during the pro-metaphase, these kinetochore areas are bound with the dyneins, and they are trying to move towards the minus pole. Whereas along with that, this kinesin is also bound, which is trying to move towards the plus pole. So one of the important things, what is happening in one condition and one end, there is a shrinkage. So as shrinkage is happening, and if this is moving, it will try to move in this direction, these chromosomes. On the other hand, see that what is happening right here, although the dynein is trying to move towards the minus end, but what is happening right here, there is a constant growth of this microtubule towards the plus end right here, and the kinesin will try to go on the plus end. So here it is a pull by this dynein. Here it is basically a push by this kinesin. So there is 2:1 dynein trying to go at the minus end. So this loses the game. So here, this dynein and the kinesin, I mean, like, this is completely theoretical, but overall, that the forces to go into the middle portion is much more, and that aligns the chromosome in the middle portion. So that's basically the idea. So kinesin 4 is really not needed. This kinesin helps in the condensation of the chromosome. That's again, I'm not going to ask that. So just know that this NDC-AT complex, like a sleeve, which is a part of the outer kinetochore, that is important, and how it is connected to the inner kinetochore. And the most other important part is that how the regulation of the bioorientation happens. So when these are close together, remember that, again, make sure that you remember the CPC, or the chromosome passenger complex, that these CPC passenger complex contain this aurora B kinase. And when there is no tension at this point, they are close together. And this phosphorylation of these NDC complex right here doesn't allow many of the microtubule to bind to them. But as the more and more microtubules are growing here towards these NDCs, I'm sorry. So right here, what is happening that when you have this low tension and these microtubules are growing and growing, some of these not phosphorylated NDC complex will bind. And this is just random because see that aurora B kinase is phosphorylating most of them, and there could be some of them which are free will bind to these microtubules, just a few, as it is going towards the prometaphase. And these microtubules will try to pull this area apart. And as they are pulling apart, gradually these aurora B kinases will be much more distance from the NDC, and more of the microtubule will get recruited, and that will separate these two out. So that's the process. The more happens, more this high tension happens to move these towards the middle. So just the treadmilling that happens, just know the fact, and you can just use the fluorescence tag tubulins to track those. That's straightforward. And just know that what is happening in anaphase A and the anaphase B, that how these are working. So one important part is that here at this time, right here, these kinetochore microtubules are shrinking, pulling the chromosomes towards the spindle fiber, whereas here you can see the polar microtubules which are overlapping are pushing each other. That increases the distance in between the poles, and that will elongate the cell as well as the spindle fiber. So that concept basically. And let's go into this one. That's what is happening. You can see right here that this is the pushing of the two overlapping region of the two polar microtubules, whereas you can see that right here, the dyneins which are connected with the cell plasma membrane, they are trying to just walk towards the minus end. Remember that dyneins are minus end going motors as they are trying to go in there, but these microtubules plus end is trying to grow. That increases the tension and the plasma membrane is stretching. So some polymerization also goes on so that the cell can grow in length. So these are just some of the idea that how the different types of movements are happening. Right here, remember that, as I said, that kinetochore microtubules are shrinking here. As there is a shrinkage happening at that time, on the other hand, the kinesin 13 will try to just move. So they're at the minus L, and automatically what happens that if they're trying to go towards the plus end, it will automatically just get pulled towards the spindle. So here you can see that here is also the shrinkage at the minus end. So minus both area, plus end as well as minus end, shrinkage happens to both end, right here as well as right here. And here you can see the polar microtubule, how they are being pushed to each other. The kinesin 5 is a dual-headed motor, so it is pushing towards the plus end, this one, and it's also pushing this one towards the plus end. That helps. And of course, the pulling of the dynein, which is towards the cell periphery, and it's trying to go towards the minus end. Remember that the dynein is trying to go towards the minus end, which is the spindle, whereas the microtubule is growing towards the plus end. So this is going this way, this is going this way, and it increases the tension to stretch the plasma membrane. So these are some of the concept, I guess. And let's see what's the next part. Cell cycle part 2. Just remember the checkpoints, where the main checkpoints are, how the scientists discovered the mitotic promoting factor. And again, remember that the cyclin and the cyclin-dependent kinase concept, remember that you have a specific cyclin which is synthesized and degraded in every cycle and the step, whereas your cyclin-dependent kinase or the CDK level remains constant. But upon binding to the cyclin, it gets activated. When it's not bound or dimerized, it gets degraded. I'm sorry, when cyclin get degraded, the CDK will not be active, but the activity of the CDKs are always high or always the same throughout the cell cycle. That important concept. So this is just an overview of that. So here you can see the cyclin-dependent kinase level, this doesn't get degraded. Only the cyclins in each and every step gets degraded. That's an important concept. This gives you the overview of that. And remember that your APCC, this is an ubiquitin ligase, so that the mitotic CDKs can get deactivated or degraded during the mitosis. So that's important. So yeah, I mean, like, this is the complex part of it. So remember that what is happening in each step, that how the G1 CDK cyclin kinase is activating these proteins. Now remember that your S-phase cyclin CDK is this complex. This is synthesized in this step, but it doesn't allow to go to the next step of the S phase due to this inhibitor. And SCF, this allows this inhibitor to get degraded so that it can just go into the S phase to do the DNA replication. So it's synthesized in the previous step, but not activated until. And so these are some of the concepts. This is a little bit detail about the cyclin D CDK4 and cyclin D CDK4. Remember that CDK4/6 is not a single thing. So that is an important concept. Remember that this is actually two different things. Cyclin D with CDK4 and cyclin D with CDK6. These will activate the RBE2F complex, phosphorylate the RB, where RB will separate out from the E2F, and the free E2F will allow the gene expression. And also the cyclin E CDK will also help in this phosphorylation of this RB. This is just another rendition that what it does. So once the cyclin EA CDK2 complex, which is right here, will be activated, that does allow the S-phase proteins as well as centrosome duplication that happens. That's the same rendition of that. That's the tumor, why the retinoblastoma is named like that. So remember that if the retinoblastoma is mutated, at that time, the E2F is always active. Remember that. That is the very important part. So we can just, this is a very good question that we can just ask some questions that you have, particularly a patient where the retinoblastoma gene do have a mutation where the retinoblastoma cannot bind to E2F. And straightforward from there, if it cannot bind to E2F, E2F is constantly going to be on. And that's the reason that it will undergo uncontrollable cell cycle. That's the outcome. Remember that if E2F is always active, the cell cycle will not stop. It will go on, and that will lead to the cancer. So know that, and definitely we'll have some questions on that. Oops. And here, just remember that the inhibitor that we talked about, that is the SYK1, which is phosphorylated which is phosphorylated. This is your phosphorylated form, and at that time it cannot be activated. But remember that your G1 cyclin CDK complex, which is right here, you know that what they are right here. And these G1 cyclin CDK activates the S-phase cyclin CDK components, and how they do this. Remember that right here, they will initially phosphorylate that SYK1 complex. Once it is phosphorylated, then if you go back right here, this SCF proteasome will degrade this inhibitor once it is phosphorylated, which is the SYK1. So this is your important part, that it will be degraded once it is, and now the S-phase cyclin CDK complex is active for DNA replication. And here again, just remember the ORC, that this is the origin of replication, and you do not need these individual names of these proteins at all. So just remember that your S-phase cyclin, so your take-home message, your S-phase cyclin will phosphorylate a number of these proteins which are inhibitors and bound to the ORC, doesn't allow DNA to replicate. So these need to come out, these proteins which are inhibitors need to come out of the ORC from here so that there is a replication complex can form. So see that the ORC is basically blocking the origin of replication. So this is a block. These are all the things that is actually blocking. But what you are seeing that at this point, there are some other proteins which are the helicase not needed, but they are being loaded, but still it's not done. But here what you are seeing that as these portions are coming out, these are all together inhibitors. As they come out, there will be a bunch of other proteins that will activate and phosphorylate these complexes to undergo replication. Very general term. So just remember that the inhibitor complex which blocks the site of the origin of replication is removed by the S-phase cycling by phosphorylation. That's your take-home message. Not detail is not needed. So here, just remember first two things. Again, just I will just carefully tell that exactly what you need for that. Sometimes it may look like that is an intimidating slide. So first thing that you need to know that you have 3 specific checkpoints, which are non-DNA damage checkpoints. So again, 3 specific non-DNA damage checkpoint. Number 1, your unreplicated DNA checkpoint. Remember that. This is activated by the ATR and check 1, which inhibits the CDC25C. And that will inhibit the cyclins. The second one is the spindle assembly checkpoint, which is activated by MAD2, which will inactivate the polyubiquitination of securin. What this protein does, is everybody good? Can you see the slides?
Yeah.
Okay. Yeah. So right here, what is happening, that securin, remember that we saw it in the last part, that the 2 sister chromatids are attached with this securin, or the cohesive, they are bound together with this. So if the 2 chromosomes are not, sister chromatids are not being able to separate by this, they will not go into the mitosis. So that's your spindle assembly checkpoint. Then chromosome segregation checkpoints is through this. This essentially is, this complex doesn't allow the polyubiquitination of the B-type cyclin, and the inhibition of this is caused by the chromosome segregation. So if it is not properly aligned onto the metaphase plate, the cell cycle will halt there until the chromosome go to each of the poles properly. So these are the 3 things, mainly overview of that. And then you have the 3 DNA damage checkpoints, and this is much easier to understand as such because all of them works the same way. So it works through this ATMR complex, which activates the P53. Absolutely remember that. That is called the guardian of the genome, that complex, which is very unstable. But so it constantly being produced in the cell and it gets degraded. But if the ATMR, these protein complex, which looks for DNA damage, is activated, it stabilizes the P53, which will activate this P21 CYP. And this P21 CYP can again work on all the different or block the activation of all these different cyclin at different stages, and that will block that cell cycle. So this is the important part. Here, one of the important thing, remember that this is underlined right here, that these S phase entry, or right here, that after the S phase, that means that all the DNA before replication is damaged or not, that is checked here. And that's the reason that this is very important. And check 1, 2 helps the CDC25 right here, blocking that. It will block the entry to the S phase. So if the DNA is damaged, it will not allow. And that's why you have a dual checkpoint right here along with this, because it's a very important checkpoint. The next one is also important, and that's why you have a dual control through the check 1 and 2. After replication of the DNA, if the DNA is damaged during the replication, then the DNA will not also enter into the mitotic phase, into the G2 phase as well. So that's why these are the 2. So entry into the S phase, exit from the S phase into G2 is both checked by this dual control. So just remember that part. So this is basically the activation of the same process. And I think that's pretty much it. So you guys think that you guys are good with the last 2 parts? I think that those 2 parts are super easy. I should not probably go over that. So do you guys have any other questions that you can think that you can ask?
What do you think the biggest format changes? Are there going to be significant format changes between this test and previous tests?
No. It will be there will be not much significant format changes. So the thing is that what you guys do is like this is how it's going to work. That there probably will be a little bit more questions in the question bank, so that just to maintain the integrity of the questions, and there will be some questions which are, so basically what I will try to do. still probably 70% of your questions will be straightforward questions that this is what it is, this is what it is. Like if you, that basically recap what we call as like your Bloom's taxonomy 1 and 2. So MAP kinase does this, G protein does this, upon binding to a ligand, the alpha beta subunits separate. So just straightforward memorizing these small things, those things will happen. And do practice your practice quizzes and the quizzes that you have for both. And many of the questions you can see from there. Some of them may change the choices just a little bit, and just be careful with that. So just don't remember that the choice was right there and answer the C or D the same way, just memorizing those questions. Just read them carefully. So those are the things. And there will be probably about what I'm trying to think that I will try to reduce the number of critical thinking questions to say not more than 20%, so that you have at least 80% of the question that if you read and know the material, you will be able to answer that. And so I will definitely reduce the rigor. So I have already reduced the material from the 3.2, whereas someone asked that 3.2 part 1 and part 2 is pretty much detail. Again, although it looks like a little bit detail, but still the original material is much more than that. But that is one of the central part of the cell cycle, very important to this course. That's why it is a little bit more detail. So that's about it. So I will say that I will definitely reduce the rigor of the course. And the thing is that if you have studied the way you studied at for your test 1 and 2, if you study at the same level, you should expect a grade increase because the content is a little bit easier, and I will be a little bit lenient about asking too many difficult questions or convoluted questions. So the critical thinking questions, there will be some questions that will be challenged. You will be challenged with that, but the number of them will be a little bit less. So that's pretty much it. But definitely, I'm going to post all the questions for your clicker. So look at those clicker questions. I have not posted that. So I will post that, and definitely do your practice quizzes over and over. So you must know all those questions for sure. Make sense?
Yeah, thank you.
All right. Any of you, any other questions? No? Everybody good? Okay. Thanks a lot, guys.
Thank you.
Yep, sure. So I will just keep the, so we are not meeting, of course, next Tuesday. If you guys have any questions, just shoot me an email. That will be great. Yeah. So don't forget to do that. All right. Have a good weekend, folks. Do study. Utilize that time. All right. Bye-bye then.